Viewer discretion is advised.
That video gets me every time. Welcome to channel PLB 112. Plants are incredible organisms. They have the ability to grow from a tiny seed into a towering tree with a little help from a process called endoreduplication. So what exactly is endoreduplication? We actually have Jeremy here in the studio today, and he'll go ahead and explain that for us. Take it away, Jeremy.
All right, thank you, Justin, for that introduction. Now we're going to explore the question that is on all your minds. What is endoreduplication? Well, endoreduplication is a process in which a plant cell undergoes multiple rounds of DNA replication. However, it doesn't undergo cell division. Due to this, the cell is left with an increased amount of chromosomes. With this increased amount of chromosomes, the plant cell becomes polyploided. Now to take a deeper look into this, we have our beautifully drawn plant here. And we're going to look at a little segment of that plant. We're going to see all the little individual cells in that plant. And now we're going to go ahead and isolate one of these cells. And now this single diploid cell is going to undergo endoreduplication. And with endoreduplication, that diploid cell can undergo two pathways. One of which is called endocycling. In endocycling, this diploid cell is going to result in one big nucleus, or one enlarged nucleus. And the other pathway is called endomitosis. And endomitosis results in two or more nuclei in a single cell. However, endomitosis is special because it can do what endocycling does as well. So an example of this would be these two nuclei end up being an enlarged both getting enlarged, so it resembles the endocycling process. Now to completely understand what's going on in endomitosis and endocycling and endoreduplication as a whole, we had to take a deeper look into the mechanisms. However, that is will be explained in a later portion of our topic here. I'm gonna go ahead and pass it back on to Justin.
My boss, he's I don't get paid enough for this job.
Justin, we're live.
It was at this moment that he knew. He f***ed up.
Thank you, Jeremy, for that segment. That was a little difficult, so if you guys didn't catch all that, I'll go ahead and I'll summarize it for you. Endoreduplication has two pathways. The first being endomitosis, and endomitosis could lead one of two ways. The first being it could have one enlarged nucleus, and the second, it could have a multinucleated cell. The second pathway it can go is endocycling, and endocycling will result in one enlarged nucleus. Now we're gonna go on break. Once we come back, we'll have a special guest for you guys. Go ahead and enjoy the break.
It's nice to rest your eyes and maybe go outside. Grab a book and get some air. So what are you waiting for?
Welcome back. Hope you enjoyed that break. Today we have a very special guest here in the studio. His name is Dr. Pirana.
Well, thank you so much for having me today, Justin.
Of course. So Dr. Pirana, can you go ahead and explain some of these mechanisms in endoreduplication for us?
Oh, no. Well, Justin, I'm very late, actually. I have another lecture to attend. So you could just watch, I uploaded a video on that platform. What's it called, Justin? YouTube. And you can watch the video, all right? Yeah, I'm very late. Thank you so much for having me, Justin.
I guess we'll just play the video now.
Hello, I'm Dr. Purana, a 5-time Nobel Prize winner. And today, I'll be discussing endoreduplication. But before we do that, let's go ahead and take a look at normal cell that undergoes mitosis. Before the cell goes into mitosis, we have the G1, S, and G2. Then we go into mitosis, where we have the pro, meta, ana, and telophases. Simultaneously, the cytokinesis, where the cell will go ahead and split, and we start over again. So in a normal cell, we have a 2 DNA content in a diploid cell. Eventually, after it goes to S and G2 phases, the cell will grow into 4C and still a diploid cell. But once it goes into mitosis, the DNA content will go into the opposite poles, and eventually will form the cell wall plate, which will, through cytokinesis, will eventually be the cell where the cell wall will be, and the cell will split, leaving us with 2 diploid cells that have 2 DNA content in each. Knowing that now, let's go ahead and talk about endoreduplication. Well, first of all, there are 2 types of endoreduplication in plants. We have the endomitosis and endocycling. A cell that undergoes endomitosis or classical endomitosis can end up in 2 ways. The first way is that the cell will form as a polyploid with multiple nuclei. Each nucleus will contain 2 DNA content and will be diploid, leaving the overall cell having 4 DNA content and a tetraploid. This happens when the cell during mitosis only goes up to telophase. The second way is that the cell will form as a polyploid with a 1 big nucleus, having multiple DNA content, leaving the cell as a tetraploid. This happens when the cell during mitosis only goes up to anaphase. A cell that undergoes endocycling, on the other hand, goes through a process that only contains G and S phases. Now, G is actually a modified G that contains a fused G1 and G2, and a cell that undergoes endocycling does not go through mitosis. So if we start with a diploid cell with 2C, eventually it will go through the growth phase and it will replicate its DNA content, having 4C and still being a diploid cell. And since the cell will keep going through the G and S phases over and over, it will keep on replicating its DNA content, leaving us with a huge nucleus having many DNA content. Now let's quickly summarize key differences between both endocycling and endomitosis. Endocycling forms 1 big nucleus. It alternates between G and S phases. However, endomitosis can form 1 big nucleus also only if it goes up to anaphase, or it can form multiple nuclei if it goes all the way up to telophase. However, both of them don't undergo cytokinesis, and only endomitosis goes through partial mitosis. Make sure to like and subscribe for a chance to win one of the iconic Purana hats. And I'll see you in the next video.
What a great video by Dr. Purana, but honestly, he was a little weird.
I wouldn't say that. I thought it was pretty cool. He even gave me one of his hats.
Anyways, let's just go ahead and jump into the biological significance of endoreduplication.
That's interesting because endoreduplication actually has a lot of biological significance, one of which is the cell enlargement, which was discussed earlier in the segment, where the cell enlargement actually helps with the growth of the larger structures in the plant. Also, it's a natural defense against the pathogens.
You know, I've actually read that it could help with DNA repair by increasing the number of copies of DNA.
That's interesting. I haven't really come across that in the readings I've done.
And actually, I do know that it is a part of a lot of real life examples, such as like cucumbers, corn, tomatoes, trichome formation. It's just a fascinating biological process. Anyways, I'm Justin.
I'm Jeremy.
And thanks for tuning in.